This is the fourth video related to repetitive loads and their effect on soils. Contrary to the previous videos, I would like this time to emphasize not so much on the granular skeleton, but on the fluid. And we're going to see what are the implications of fluid cycles, on chemical transport, on heat transport, on the possibility of transporting grains, and then some barometric pumping. In all these cases, the human body becomes a wonderful analog, an inspiring model that we can also transport to the case of soils. To recap previous presentations, we talked about the fact that repetitive loads are always found in natural systems as well as in our engineered systems. So we should pay close attention to the potential long-term impact of a large number of repetitive loads. In the case of stress cycles, we recognize that from a volumetric point of view, it's going to converge to an asymptotic terminal density. From a point of view of shear, the shear strain accumulation could arrest in the case of shakedown or continuously adding shear strain in what is called the ratcheting mode. In both cases, analytical and numerical analysis were presented, where numerical analysis basically are required for more complex boundary conditions. Then we talked about the multiphysics type of repetitive loads, and we recognized that coupling mechanisms were intimately related to grain size in most cases. For example, in chemomechanical coupling, for grains that are micron and submicron, the clay materials, the thermomechanical coupling was surprisingly grain size independent, and we actually demonstrated the effects with big rock blocks. Once again, we went back to scale dependency for suction cycles and gains relevance in sub-millimeter sediments, including the silts, particularly the clays, and we show how important free thaw cycles could be, particularly when freezing produces grain displacement, which is going to be very important in the finer materials and at lower confinements. Finally, we are reaching today's presentation, which is going to place emphasis on the fluid flowing in pores. So let's start. This is a beautiful story. During a presentation in the 1860s, researchers showed the cross-sectional look of a femur, and Kuhlmann, who was present in the audience, recognized that the cells tend to follow a characteristic pattern, and that characteristic pattern resembled the structure of cranes, which is what Kuhlmann was designing. This is the Kuhlmann that gave rise to the analysis of trusses, which is Kuhlmann's method. And so he thought of the bone, the head of the femur, looking like the design of a crane. So this was further confirmed, but therefore we understood right away that the bone grows in response to the stresses that are applied. Now the question is why? If it was diffusion control, there wouldn't be enough time for nutrients to reach the bones to support bone growth. So now in 1974, so 50 years ago, it was recognized that actually diffusion was not enough, that the action of squeezing and relaxing a bone has a very important effect on bone growth and the response to stress of cellular growth. Now, how could that be possible? Let's go back to understand this from a very simple case of a capillary tube. Imagine that I have a red fluid and a blue fluid, and if I advance the red fluid and then I bring it back, the blue fluid regains territory, and this can go back and forth without any implications. In other words, there is theoretically, under these conditions, no mixing. In practice, there is some mixing. Now, what happens if the tube is connected to pores? And so there are these channels connecting pores. In the case of bones, these are called the canaliculae, and these are called the lacunae, little lakes and little canals. And if we follow the same example, once again, there is zero net flow at the end of every cycle. The integral of the flow rate gives us zero, the integral in a cycle. However, when the red fluid advances and reaches the pore, when it comes back, it leaves some of the red in the pore where it got mixed with the blue. So now when the blue pushes the red to the left, it mixes in with some of the red left behind at the pore, which then gets transported in the next cycle. So even though there is zero advective fluid flow at the end of the cycle, there is species transport in this process. And why is that? Because there was advective cyclic flow combined with mixing at pores. So this is indeed the case of bones. If we do the mathematics very carefully, satisfying continuity conditions and mass conservation, solute conservation, and we take certain asymptotic conditions to simplify the analysis, in which we assume that the total volume of flow in each cycle going in and out is much greater than the volume of the canaliculae, and in fact that the volume of the pores, of the lacunae, is greater than the volume of the canaliculae, then the mathematical expression converges to the diffusion equation. So effectively, this back and forth with mixing in every cycle, effectively, it's a diffusive process, but with an accelerated diffusion coefficient. And what is the effective diffusion coefficient? Is the length scale of the system, which is the center to center of each lacunae, square, divided the period of every cycle. And it is magnified by the volume of flow going in and out compared to the volume of one lacunae. Therefore, the more fluid invades in every cycle, the fastest this diffusion process becomes. What an elegant, simple equation. Let's test it. And this is a Haley-Shaw cell, two plates, glass plates, filled with glass beads in between, and we inject blue fluid in and out, in and out. There is zero advective flow at the end of every cycle. However, as you can see, at the end of 60 cycles, the blue front has advanced, 400 cycles has gone further, 1,000 cycles has gone further. And now if we plot the progression of the front and we compare that with respect to how far it should have gone by pure chemical diffusion, we recognize that in order to advance an equal amount by regular diffusion in the order of 10 to the minus 9 meters squared per second, then it would have taken orders of magnitude longer time. Indeed, let me do it with my hands, cyclic fluid flow with mixing enhances species transport. Now, I have deviated in this analysis from what is really happening in a bone. In a bone, every time we walk, we squeeze the bone like a sponge, we squirt fluids out into the osteocanal, which is the one that carries all the nutrients and picks up all the waste. And then when we relax the weight, it sucks the fluid back in, and that is where mixing is taking place between the canaliculae going to the lacunae and the central osteocanal of the bone. Now, that's a different boundary condition. So if I now take into effect and take into consideration the fact that the bone is a sponge that is driving fluid flow, and we do a Biot-like analysis, I come up once again with an equivalent diffusion coefficient with a slightly different formulation, whereas you see the change in the volume of the lacunae, but still anticipating a very effective transport of a species with zero advective flow at the end of every cycle. Now, what are the implications for that? Well, it means that we can transport lots of reactants by cyclic fluid flow, and that can be very important for biomediated chemical reactions in soils like MICP, or it's also very important for corrosion processes. And on the right-hand side here, you see the mass loss of metal coupons buried in sand if it was totally saturated or totally dry, or air dry. But what happens if there is cyclic fluid flow? Please notice that 15 times difference in the scale. When there is cyclic fluid flow, we bring the oxygen that will support the corrosion, and we remove the corrosion byproducts dramatically accelerating corrosion. Therefore, where the water table is oscillating is where we should anticipate the maximum corrosion, and indeed that is well known and proven here with this data, beautiful data by Dr. Gloria Castro. Now, we can not only transport reactants and chemical elements in this cyclic flow, we may also transport heat. And so here is an example. Imagine that I have a container with saturated sand, and I inject hot water from the bottom to advance a certain distance L. And then I pull it out. But when I pull it out, the particles have taken some of the heat, depending on their heat capacity. And so when I push the water out, it will leave some of the heat behind, and the water that advanced downwards now is going to pick up the heat. So when I inject hot water back again to a depth L, the water that had been replaced now gets moved up and carries heat upward. So all of a sudden, I begin to realize that I can have heat transport by cyclic fluid flow. Let's see mathematically what this is all about. Then we have that at any moment, I'm exchanging heat between the particles and the water related to their masses and their heat capacities. And when I write this equation, I expand this equation, which is just the analysis of heat conservation at the particle level. When I expand it, I realize that basically I have the diffusion equation in finite differences, where the diffusion coefficient is this factor here, which I'm going to explain next. So once again, I start with a heat conservation at the particle level. I expand it. I realize that it is the same as the finite difference solution of the heat transport equation, diffusive heat transport. And these two become equal when the equivalent diffusion coefficient in the cyclic motion is equal to the length scale of invasion squared, this is L squared divided the period of every cycle, and it is related to the water content and the heat capacities of the materials involved. Another very elegant solution, and let's test it. And when we do test it, this is the red shows the predictive temperature profiles in a column where we are injecting and extracting water. The cycles look not very nice because we are stretching the scale in time, but the cycles were actually a constant period cycle. Indeed, there is accelerated heat transport, which is mathematically equivalent to diffusion. Now, building back on the metaphor, the biological similitude with our bodies, we recognize that the heart doesn't pump like a peristaltic pump continuously. It actually pumps cyclically, and we recognize that the pressure has two characteristic pressure levels. So it accelerates the fluid flow when it pumps it, and then it withdraws it, and accelerates it and withdraws. So what can that do? Well, that is pretty much the way we swim. In other words, we do certain motions very fast, and then we return slowly. So we're playing with the drag forces as we swim. And we wonder whether we can use this to transport particles, like making particles swim in the porous medium. So let me explain what we are doing. This is an experiment in which a tube is filled with water, and with a syringe, we pump water in and out, but we pump it in fast and we withdraw slowly. So every time we pump it in fast, the drag force moves the particle forward. When we withdraw the water slowly, the drag forces are not sufficient to move the particles compared to the frictional resistance of the particles against the base of the tube. And then in multiple cycles, I advance the particles, then I withdraw, I advance, withdraw, inject very fast, withdraw. And the reason why you see many lines is because we're tracking many particles. At the beginning, they were all bunched together at the entrance, and they begin migrating relative to each other at different velocities, which is also a form of optimizing the drag forces in how they interact. Imagine these are like cyclists following each other. Finally, let me show you another example, which is how we breathe. Every time we breathe in, the air comes in into our lungs, pretty much the relative humidity of air, which in an office space is probably 50%, or in a cold winter night with heating in a house, it could be as low as 20%, very low. But when we breathe out, air comes out fully saturated with moisture, because when it reaches the end of their pathways in the lungs, it reaches the alveoli, which are these little sockets where there is fluid air interaction, where the blood gives away the CO2 and picks up oxygen. It also releases moisture. And so the air that comes out is pretty much 100% saturated. And so we are losing weight by breathing in dry air, breathing out moist air. We reproduced that in the laboratory, doing exactly the same boundary conditions, and we were monitoring with a nuclear magnetic resonance device, so we were not perturbing the conditions inside the chamber, trying to pick up the evolution of moisture. And by the way, this will be equivalent to how the atmosphere interacts with the vadose zone, this unsaturated zone, near the surface. So it gets pressurized and depressurized, and so there is breathing of the subsurface into the atmosphere. So let's see what happens. And indeed, we are losing moisture as time passes, starting from initially quasi-saturated conditions and eventually draining all the water. So if we look at the cumulative water, is indeed evaporating the water effectively very fast. In conclusions, once again, I would like to insist that repetitive loads are everywhere and affect all our natural geosystems and engineered geosystems. It is affected by hysteresis and apparently by nonlinearities, like we saw today with non-symmetric, almost like a beat function in the heartbeats. Cyclic fluid flow enhances heat transport, the transport of a species, the transport of nutrients, of vapor. And if it is asymmetric, we showed that we can even accomplish the transport of grains. And not mentioned today, but now you can relate to the previous presentation, it can trigger hydrothermo-chemomechanical couplings, because as particles migrate, because either we remove particles or we add particles, changing hydraulic conductivity, which will change the response on effective stresses, etc., and chemical changes will do the same. The implications of today's presentation, which are the consequences of cyclic fluid flow, are everywhere, from barometric cycles, saltwater intrusion, energy storage systems which will be compressed and decompressed, corrosion effects, biomediated reactions, etc. We show some closed form estimates for simple boundary conditions, and surprisingly, in several of these cases, we concluded that cyclic fluid flow becomes an accelerated equivalent diffusive transport. In more complex cases, we will need to run coupled process simulators with the proper boundary conditions. Some of the references for these studies are found here, and new papers are coming out. Don't hesitate to reach us.